All right. Hi, everyone. Happy Friday. Welcome back to this week's Single Cell and Exploration Club, where we highlight some of the features on our platforms and try to showcase them in the form of a case study. So for this week, oh, I'm sorry. For our introductions, my name is Julie and this is Liam, and we're both scientists here at Balturing. And so for this week's SCSE Club, we'll be visualizing epithelial and neoplastic cells and how they co-localize in colon cancer. So this is kind of a continuation from our story from last week where we examined gastric cancer. And so in our previous episode, we found that in normal tissue or in normal cells, we see a large proportion of pit mucus cells. But as we transition into an inflammatory state, to a metaplasia state, to cancer, we see less and less pit mucus cells and actually more metaplastic stem-like cells. And so we hypothesize here that this loss of mucosal cells, mucosal epithelial cells, could be driving the gain of cancer stem cells as well. And so, in a sort of similar vein, instead of examining gastric cancer in the stomach, we'll be looking at colon tissue instead. So, a little bit of a background on the relationship between epithelial cells and cancer cells. If we look at this diagram here, when we start in the primary tumor in colon, these epithelial cells can start to lose their cell adhesion through the process of EMT, or epithelial mesenchymal transition. And this is where these cells will start to lose the tight junction between them, which means that when they proliferate, they will start to migrate into the bloodstream. And this migration is known as intravasation. Once they migrate into the bloodstream, they can exit and end up in a completely different tissue in the liver, for example. And so here they can undergo a process called macrometastasis, where the cancer cells will start to regain some epithelial properties. So not only can epithelial cells transition into mesenchymal stem cells, the transition can also happen in the reverse way, where mesenchymal cells can transform into epithelial cells. Okay, and for today, Liam will introduce the data set that we used in today's analysis.
All right, thank you for having me, Julie, again. So now that we know cancer cells and epithelial cells have this really close relationship, so given a tumor tissue, we really want to see if they co-localize together. And to do so, we will use spatial transcriptomics. And for this talk, we will use a spatial colorectal dataset of 10x xenome, which is targeted to 380 genes. So here are the key metrics for this dataset, but more specifically, it contains two samples, which is the normal cells. In the next slide, please. So as you can see here, this spatial dataset firstly contains the healthy colorectal tissue sample. You can see there are still epithelial lining over there, and also the globular structure that does contain epithelial cells. On the other hand, it also has the colorectal cancer tissue, which has been deformed. So today we want to find where has our epithelial cells translocated to in the colorectal cancer sample, and if they can be found also within or next to other cancer cell population. And to do so on spatial transcriptomics, we will have to do clustering and annotation of cell types. And therefore, the agenda today will be the three different methods for clustering and annotation using spatial transcriptomics to ultimately find the epithelial and the cancer stem cells. So for the first method, which is called the expression-based, in the next slide, please. So like the name suggests, it will use only gene expression. So using gene expression, it answers two questions. Firstly, which of the cells in our dataset have similar expressions? And secondly, for their expression, is there any possibility that we can predict their cell type? So this method is fairly simple and straightforward on our platform SpatialX. So in the next slide, we have demonstrate the tools that you can use for doing expression-based analysis. Firstly, we have the clustering function, which simply cluster cells based on their gene expression. So we would identify the cell cluster that have similar gene expression, and the appealing feature is the next one, which is cell type prediction. So again, using this gene expression of a cluster, it compares with existing gene expression that have been curated on the online databases. So therefore, there are some considerations for using this method, which is firstly, we should have criteria for the cell type, tissue, or conditions that we want to limit to in our prediction. And secondly, for studies such as ours, which have a defined gene panel that target gene markers in colorectal cells, this is extremely helpful as it helps to narrow down certain cell types. So in the next slide, we will demonstrate how this feature looks. You can see that we had chosen the tissue to be the digestive system. This is an umbrella term that covers many other terms such as oral cavity or esophagus, as you can see on the right panel. The reason why we include all these terms is to not create a bias towards our colorectal tissue. And we also choose some subtypes, even though we do not show here, that you can see on the right panel. We have chosen different subtypes ranging from epithelial cells to, say, immune cells. So let's look at the results, shall we? So this is our initial data set. And then using this feature, we have defined different cell clusters based on how similar they are in terms of gene expression. And then we annotate them with different cell types, as you can see on the left. These cell types are the ones that satisfy the criteria that we have set on in the cell prediction feature. So there's one great finding in this first method, which is in the next slide, where we have confined to only two populations. You can see that the cells in the yellow, which are predicted to be epithelial cells, are blending in with neoplastic cells, which points to possible co-localization of these two cell types. And because this first method is pretty straightforward in the sense that it outputs readily the cell type, we can do other downstream analysis, such as finding the gene marker that is are the genes that are up or down regulated in either one of these two cell populations, which is what we did with another feature called differential analysis, which can be found on spatial X as well. So in this analysis, we found that the gene MUC5AC, which is one of the most differentially expressed genes, is more expressed in the epithelial cells. And this finding actually overlaps with what we found in the previous podcast, which suggests the hypothesis of the epithelial cells losing its function to produce mucus substances when it transitions into neoplastic cells. Could you come back to the previous slide? I would like to mention one thing though. So this result right here is not the exact cell type, but rather predictions. And actually, we have consulted with a histology expert in our team. In fact, our epithelial cell and neoplastic cell populations here are both dysplasia. But the reason why we have the epithelial cell and neoplastic cells prediction right here is because the assumed epithelial cells are actually early stage dysplasia that still retain epithelial properties, whereas the red population of assumed neoplastic cells are actually late stage dysplasia, which has the gene expression that closely resemble neoplastic cell and therefore were predicted to be one of them. So perhaps we don't want to readily assign cell type to our dataset, which points to the next method of doing cell clustering and annotation. So in the spatial data set, yes, we have the expression data, which tells how much a transcript is expressed. And we also have the spatial data, which tells where that transcript is expressed. So this method integrates both of those data to answer the question of which tissue has the same gene co-expression or genes that are together up or down regulated in the same spot. If the tissue has the same gene co-expression, it means chances are it has a distinct biological property and perhaps made of a certain cell type. And Julie will now explain how this method works.
Yeah, so as Liam explained, to reiterate, the region segmentation you can think of as very similar to Louvain clustering, where in Louvain clustering, you would cluster cells together based only on their expression profile. Whereas region segmentation not only clusters cells based on their expression, I'm sorry, not cells, genes. They will cluster genes based on their expression profile, and not only on the gene expression matrix, but also on the spatial coordinates as well. And we do so by using what is called the Moran's I-score. So if we take a look at this diagram, the Moran's I-score ranges from a value of -1 to the left to +1. And if we assume each of these squares is a different gene, for example, and the grayscale refers to their expression levels, we can say that this kind of dispersed pattern of gene expression, while it looks random, is still considered significant because while it may look random, the pattern is still dispersed and is how we expect. However, when we move to a lower Moran's I-score, the score reflects how random the expression is here. Over here, if we move to the positive side, this may be a little bit easier to understand. So we group genes together by how they're co-expressed, not only in their expression, but also spatially as well. So you can see these group of black genes here are grouped together because they can have a very high expression together within this region. So whenever we're talking about region segmentation, we only care about the positive side of the Moran's I-score. Okay, and to move on, Liam will explain some of the results we have from our region segmentation.
Right, so unlike the first method where we had different cell types, now we have segments of cell populations. These segments are created by assessing the gene expression and where they are expressed or spatial data. So for example, we have the segment number 2 that is colored by green. You can see on the figure on the left that it closely aligns with what we found to be the epithelial cells in the previous methods. And on the right panel, you can see there are 3 genes. These are the genes that are co-expressed in these segments. And by doing literature research, we found out that these genes are the marker genes in colorectal epithelial cells that are transitioning into cancer cells. So for the segments number 0, which aligns with what we found as cancer cells in the previous method, you can see that there is a bigger gene panels up to 70 genes. So to understand what these genes mean, the Spatial X platform has also offered the pathway analysis feature, which is the icon on the right there. So using this feature, and by setting, you can see the database here as rare disease. We have different databases, but just for the sake of simplicity, we confine them into only rare disease. We found out that the segment number 0 are likely to be associated with familial colorectal cancer, which have 14 out of 20 matched genes. So a nice thing is that both of the previous method and this method points to a location in our tissue sample, which is the middle part, that are likely to have the co-localization of potentially epithelial cells or early stage dysplasia and also cancer cells. So until now, we have been using expression and spatial data. What if we use another type of data, which is the last method? So in spatial transcriptomics, beside expression and spatial data, we also have the image itself, which could be a very valuable source of information. And perhaps by looking at the image, we can find tissue regions that look the same. If they look the same, chances are they have the same cell types. So this method is like having a histology expert on your team that can look at the picture and decides what's going on immediately. So this method is fairly straightforward too. It's just a simple image segmentation feature that groups similar image types. But again, this method does not output subtypes or pathway analysis. So users should have domain knowledge or a histology expert on their team to pick up the insights from this feature. And the way this feature actually works is in the next slide. So you have on the left the histology section, right? This feature will break it down into different pieces or image tiles. And not only does this feature consider the tiles, but it also consider the neighboring tiles. And actually, this feature is a foundation model that has been trained over and over again over different histological images. And given the image ties and their neighbor, it can groups ties that are likely of the same subtype. So on the right is the output for the picture on the left when subjected to image-based clustering. And actually, Julie is going to demonstrate how this works on the website.
Yeah, thanks, Liam. So image segmentation is our newest feature that we released on Spatial X. So here again, we're looking at the same image. To pull down the tools, you'll need to hover over the analysis and click on that, and you'll see the image segmentation pop up. Now, this image segmentation feature will only pop up when you have an H&E image. So again, keep in mind that this will not appear if you have IF images, only for H&E images. Once you click onto image segmentation, you can define the resolution. But for now, we can leave it as default 0.5 and then adjust as we need once we click run. Okay, and so I can show you a previous result, which was in the slide. So once you click run, the segmentation will appear over on top of your H&E image like so. And this will appear in the region layer here. And you can see how it's segmented into different regions. So with the help of this segmentation tool, we hope that we'd be able to support histopathologists in identifying important regions of interest so that, you know, people on the team, other people on the team, bioinformaticians or data scientists, can look further into those regions, examine the transcripts and examine the cell types and do some further analysis. Okay, back to our slides.
Yeah. That looks very beautiful to me, but I have a question though. What if I do not want to do image segmentation on all the tissue, but I am actually interested in a spot which you can see on the slide, I believe.
Mhm.
So you can press the arrow to the right and it should show an animation. Yes, this spot right here is what we have been what we have found in previous methods to contain perhaps the transitioning of epithelial cells and cancer cells. Can you somehow like pick out a certain cell spots and find other similar cell types? I believe you
Right.
can do this too, right?
Yes, exactly. So on Spatial X, there's two facets to the image segmentation tool. So there's the way where you can segment the entire image and it will populate the entire image automatically for you like this. However, if you're only interested in a specific region, let's turn off this region layer. So as Lee mentioned, we're interested in this area. Down here, you can click the lasso tool and you can draw on the H&E image to identify your specific regions of interest. And so if we circle that and we click search region here at the bottom, this will basically try to search for regions that look very similar to the region that we just lassoed. All right, let's zoom out a little bit. Okay, so here you can see that we have a heat map. So the yellow corresponds to uh, areas in the H&E image, which are very similar to the areas that I just lassoed. Down here, you can also define the threshold. So let's say we want to exclude some of these lower expressing regions. You can define the threshold like so. So that looks good. And you'll click Fine Tune Region. Once you fine tune the region, again, it pops up in the region layer. And here you can click Confirm and Save Region. So I'm going to save this as my region of interest. And then I can name this my epithelial cell, right? And then I'll click Confirm and Save Region. Once you do that, if you hop over to the metadata panel, you can see how we have split the metadata into cell and into region. So we're going to go over to the region, and I saved it in my region of interest here, our epithelial cells. Okay, so now once we have our regions of interest, the next question we can ask is, okay, well, what cell types are involved in that region of interest? So from our region layer, we can also transform it into our cell layer by clicking this button next to the metadata called Save to Cell Metadata. I'll click on this, and the cell metadata name I want to enter is, again, regions of interest. Okay, then I'll click Save, and it should appear in my cell metadata down here. Okay, let me try saving that one more time. Regions of interest. Yep, save. Okay, perfect. Yep, so you can see here how it switched to the cell metadata tab. And now when we enable the cell layer, we should be able to see our selected cells of interest in blue. And the cells that we're not interested in are the cells in gray. So we can click on that to see this. And let's deselect this region as well. Okay, like so. And this restricts our view to our cells of interest only. Okay, and then back to Liam to explain our results from here.
Right. So I believe it's in the next slide. So again, this is a summary and also what we found. Once you snipped out a region of interest, you can find all the cells on that picture that are of the same cell type using image-based clustering. And selecting that cells, you can visualize them on spatial X as well, on what cell types could be there using the first method of prediction. So for our SNP region of interest, as you can see on the right, it consists mostly of the two cell types, which are the epithelial cells and neoplastic cells, according to the prediction. So again, this further conclude that that specific spot on the histology section is what explains the co-localization of epithelial cells and neoplastic cells. So in summary for this talk, we have demonstrated three methods for cell clustering and annotation. For the first method that did the cell type prediction, we believe it could be improved if we had more data set, especially on the dysplasia of early and late stage, so we can better predict complex scenarios such as this. And all three methods point to the same spot that can explain the co-localization of the hypothesized low-grade dysplasia in colorectal cells, with the more late-stage or high-grade dysplasia that have the expression of cancer cells, more like cancer cells. So thank you all for listening, and I believe we have a survey for this spatial aids talk.
Yes, thank you, everyone. Just to summarize one more time, uh, basically we wanted to demonstrate here how in Spatial X we were able to utilize all of the different components of spatial data to help us supplement our findings. So let me go back to our agenda here so it'd be easier to understand. So here in method one, or In general, spatial data includes the gene expression matrix and the spatial coordinates. So in method one, we were able to utilize just the gene expression matrix to find out the cell types, to look at the gene expression. In method two, we were able to utilize both the gene expression matrix and the spatial coordinates to help us perform region segmentation. So this allows us to look at genes that are co-expressed together, not only based on expression, but also based on their spatial location. And in the third method, we were able to utilize the image, the H&E image, to segment different regions just based off of the image or the spatial data alone. And so in that way, we try to build out these analytical tools on Spatial X to help supplement your previous findings. Okay, and now we'll take this time to answer any questions in the chat. If anyone has any questions in the Q&A as well. We'll give it a couple more seconds for everyone to finish filling out the survey. So we're really excited to showcase Spatial X. So we're interested in seeing what types of technology everyone uses. Xenium, Cosmics, Visium, or even proteomics, and maybe we can showcase that the next time we have a Spatial X talk. All right, everyone. I think that's good for today. So thank you, everyone, for joining our SCSC Club today again. And we'll see you guys next week.